Hey, we're back today. We're going to talk about translation. So, this is our PowerPoint. The follow along notes will be posted down below, and you'll be able to click on the link and it'll take you to them if you need them. I will also post the flowchart and other resources that we used in class. So, let's get started. Okay, so we're just doing a quick review of what transcription is. So, this is transcription, right? If you remember, we are trying to get from in this whole process of transcription and translation, we're trying to get from DNA to a protein. So the first we talked about transcription, which is DNA to mRNA. So that's what this little box of this process here. So transcription is making an mRNA from DNA. That is done by unzipping the double-stranded DNA, which results in a non-coding and coding strand. Now, this matching of the new mRNA nucleotide bases is done using the complementary base pairing rules to the non-coding strand. So the mRNA, which is this right here, gets its copy from the non-coding strand. So that's this strand right here. And this would be the coding strand up here. Now it seems kind of backwards that it's getting the genetic code from the non-coding strand, but that's because this strand right here is a mirror of this strand, and it really wants this sequence. So by copying this sequence right here, it gets this sequence, because it's kind of like a mirror, if that makes sense. So these are some important questions. So what replaces thymine in mRNA? That's uracil. So adenine matches with uracil in RNA. Another one is where is mRNA made from? And that is the DNA non-coding strand. So those are two important questions to remember. So now in the translation, so this is the part right here. So this is taking that language of bases and translating it into the language of amino acids to give us proteins. So the mRNA leaves the nucleus and travels into the cell's cytoplasm. This is important, right? Because DNA can't leave the nucleus while RNA can. So that's why mRNA is a key factor in this whole process because it can leave the nucleus unlike DNA. So it goes into the cytoplasm, then it'll attach to this organelle called a ribosome, which is made of rRNA, and the process of translation begins. So in translation, so this is happening at the ribosome now. Each mRNA codon matches with a complementary anticodon on the tRNA. Codon is equal to 3 nucleotide base sequence. So if you look over here, this is a small section of mRNA. Here's a 3 base sequence. For example, it's AUG. This is a codon. Each tRNA carries a specific amino acid. Now, this is the tRNA right here, this orange here. This is the tRNA. This up here is the amino acid that it carries, and this is the anticodon. So this is a 3-sequence base that's the complementary to this right here, to the codon on the mRNA strand. Now, when these match up with the right codon, it places the proper amino acid into a growing polypeptide chain, and a polypeptide is a protein. If you remember, we get our amino acids from food that we eat. So the only thing you know right now is that we get the amino acids from the food we eat. They're available, and this tRNA that has a UAC will always go and grab the same amino acid. So UAC will always grab the same amino acid. Then it'll come back to that ribosome, find the right codon to match its anticodon, and then it'll fit in, and then boom, releases that amino acid. So right here is a genetic code. So this is a tRNA anticodon and amino acid chart. So basically how this is read is you have your first letter here, your second, and your third, and it'll tell you where or what amino acid codes for that anticodon. So what anticodon codes for which amino acid? So, example, if we find our UAC like we did in that previous picture right here, look, start, thing, same amino acid. So this is our mRNA right here. CA, our TNA, see the base pairing rules still apply. U to A, C to G, A to U, right? Because we don't have that thymine. This is still RNA, therefore it's still uracil, right? And then here will be our amino acid sequence. So amino acids based on these anticodons right here. So this is an anticodon, this is an anticodon, this is an anticodon. Now this box is a little misleading because these are all separate. So each tRNA only has one 3-base sequence, which is the anticodon. So this is just some practice. I'm just going to run through this. If you want to practice yourself, you can freeze the video, work on it, and then start it to get the answer. But here's the non-coding strand, right? These are both DNA, therefore thymine matches with adenine, and AT, CG, AT, AT, so forth. So this is our mRNA now. As you can tell, right? A to U now instead of A to T, because this is RNA. T to A, G to C. So now we're going to go into translation. So we have our mRNA. Okay, we have our codons. So these are codons right here. These 3 sequences. So we have 1, 2, 3, 4, 5, 6 codons. Now these are the anticodons that match. UAC, AUG, UUC, see? So to these codons. And now we go to our chart, and you should have that yellow chart, and I'll provide that yellow chart in the links below. Alright, so it's important to know that every protein sequence, polypeptide chain, starts with this anticodon and this amino acid. This is how it knows where to start. And if you look at your chart, you'll notice there's a couple stop ones, and those are important because once it reaches that amino acid or that sequence, it's like, okay, stop right here, cut the chain off right here. We have our complete protein. So I'm just gonna go through these right here, and then stop. You see, stop. So that's the end of our chain. Start, finish. So that's it for a translation. I went through it pretty fast. If you have any questions, leave a comment. Ask me in class. I will also be trying to make a video that goes on to what happens in a ribosome to try to make that more clear. Thank you.